Hello and welcome to Preprints in Motion, a podcast taking a deep dive into the fast-paced world of preprints.
Join us as we sit down with early career researchers, discuss their latest preprint, and find out about their journey through the muddy marshes of academia.
Hit that subscribe button, leave a rating, and find us on Twitter @MotionPod. Support us by heading over to buymeacoffee.com/preprints. But for now, let's get into the show.
This week we have some friends from Sheffield, Alex and Kavitha, discussing Tanycyting and the hypothalamus.
For once, it's a full house. We've got John with us for the first time in ages.
After a long break of not being in front of the microphone and only being present in editor overlord form, I am actually back in front of the microphone.
And we'll cut all that out, so it'll be like you were never here.
I will not be cutting that out, don't you worry.
And we're joined by people we actually know today. I don't know if you're friends of the podcast, because I don't know if you listen, and I'm not going to put you on the spot, so you don't have to answer that.
I know Alex listens.
Alex listens. Oh, at least one does. But you are friends.
We are friends, yes.
There you go. We'll settle on you are friends.
Yes, that's better.
So because when we have multiple guests on, I think it's nice to let people introduce themselves, because it saves me doing it. It saves me a job. So I guess why don't you each sort of give us a little bit of who you are, maybe what you're currently doing. So Kavitha, maybe you want to go first.
Hi, I'm Kavitha. So I'm a postdoc in Professor Marisha Platek's lab, and I basically from India. Came here to do my master's, PhD, and I haven't moved out of Sheffield at all since then. So I've been stuck in this building. Perthcote is my forte, I think. So
there's worse places to be stuck, right? It's a beautiful building.
That is very true. Perthcote is very beautiful. So yeah, I am here postdocing with Marisha at the minute.
And Alex?
Hi, yeah. So I go way back with some of you. Me and Emma first met in 2010 when we did our undergrads together in Sheffield, and both continued there doing PhDs.
You just told everyone how old Emma is.
Sorry.
How dare you?
Sorry. And so did my PhD in Sheffield with the other people on the call. At the time, when I did my PhD, I was working in Marisha's lab where Kavitha now is, and I was joint supervised by Marisha Platek and Andy Furley, who are two of the senior authors on the paper that we're here to discuss today. And then after my PhD, I decided to move out, and I'm now a operational research analyst in the civil service, doing all kinds of different types of researchy things there.
Which you might be coming on a separate podcast to talk about when I eventually get around to doing that, maybe. There's a long list of things that I'm currently working on that are not progressing at all. So Alex, everyone missed the bit that we've going to have cut out, but Alex did the sensible thing and got out of academia. We're all now desperately trying to follow him out. Hopefully we'll get there. So let's start with somebody giving us a little bit of an overview as to the preprint we're talking about today, so we all understand what we're doing or what we're looking at. Who wants to do it? Who's feeling brave? Who recently read the preprint again?
Come on, Alex. Your name is first on the list.
Sure. Well, we are joint co-authors. We're joint first authors on the paper, we should point out for everyone listening. But yeah, no, so I was going to say I started this research, but I didn't start this research. It actually started based on previous research from the lab by Ian Stewart and Sarah Robbins, who are also co-authors, and then also Sarah Brown, who I've got to represent because Sarah Brown did PhD at the same time as me in the same lab, but not on this project anymore. This project was started off as part of her master's project, looking at stem cells in the hypothalamus, and to kind of take it back to the very basics, I've got to talk about what the hypothalamus is. And so for anyone out there who doesn't know about it, that's fine. It's a small part of the brain that's found in the very center at the very bottom, which means it's a really ancient part of the brain. And it sits in the center at the bottom around what we call the third ventricle, which is the third of four fluid-filled spaces in the brain called ventricles. These contain cerebrospinal fluid that flows throughout the brain and all the way down the spinal cord. And sitting below the hypothalamus is, or coming from it, is the pituitary stalk, which leads into the pituitary gland. And together, the hypothalamus and the pituitary are involved in regulating homeostasis for the body. And so this is how we keep things the same during the body, whether that's thinking about feeding behavior, or aggression, or thirst, or sleep, or reproductive aspects as well. There's many, many different things that these two parts of the brain control. And so about 20 years ago now, must be, the first study came out identifying that there was a group of stem cells in this part of the brain called the hypothalamus. And so these are cells that kind of align the ventricle that fluid-filled space in the middle. And they have these really long processes that stick out into the brain tissue around. We already knew they interacted with neurons there, and they also interact with the blood vessels in a bit at the bottom of the hypothalamus. And these specialized cells are called tanycytes. And so about 20 years ago, another group figured out that maybe there was a stem cell or progenitor cell population there, which could give rise, when prompted, to a variety of different cell types, including new tanycytes, as well as neurons and astrocytes and other cells of the brain, both in development and then in adulthood as well. And so this work was continued by other labs, including our lab. And so in this paper, what we're doing was we expanded upon what we understand about these tanycytes as stem cells. We looked at characterizing them with a variety of markers from the front to the back of the hypothalamus, because it turns out there's a lot of changes through there that aren't well characterized in the previous literature. We also identified a new marker for these tanycytes called NARCAM, which is a cellular adhesion molecule. So it sits on the surface of the cell, attaches stuff together, but also seems to be involved from our research in regulating the differentiation, proliferation, and neurogenesis that these tanycytes are involved in postnatal mice. Yeah, I think I covered everything.
I think that was good. You aimed it at my level, which is always good. And you did give me flashbacks to undergrad, so I might start and go and sit in a corner and shake for a little bit. But other than that, I think that was good.
I would have said he aimed it even at my level. Anything above, even cells I struggle with. Just give me some gooey protein or DNA in a test tube. I don't want to hear about anything higher order than that. So yes, no, very, very nicely explained.
We're still very complimentary here. So you mentioned tanycytes there, which are cells I knew nothing about. Despite knowing you for so long, I turns out I didn't ever listen when you spoke about what you did.
I read your paper and I was like, how did I not know about this? I literally known Alex since 2010. We did everything opposite each other. And
that's the thing. Hypothalamus does so much important things, and nobody seems to know what it actually does, because that's what is controlling all your main physiological everyday behavior, and people don't care about it. There you go. Here we are to talk more about it.
I mean, I hear neuroscience and I check out. It's too difficult for me. But that's a good starting point for the next question, actually. So the hypothalamus is this sort of master organ, I guess, that regulates so much and does so many different things. So where do the tanycytes that you've been looking at fit into that whole process? Are these things that are ultimately turning into cells that are involved in all of the different things that the hypothalamus can do? Or are these kind of like already down a specialized lineage?
They are a specialized lineage, and obviously they're not going to contribute to all of the hypothalamic functions. But they do play a very important role because they're mainly lined along the, like he said, it lines the third ventricle, and then it feeds into your median eminence. So they act almost like a gatekeeper cells between your body and your brain. So they are the ones who are going to communicate, sending hormones and other metabolites to your body, and then collecting the levels back and sending it back to your brain to say what's high and what's low. So they pretty much they are the barrier, and they are the interface between your body and your brain. And they can differentiate into astrocytes, glial cells, and some neurons. So yeah, that's how they regulate all these functions.
Yeah, so that was what I was going to ask next, is what makes them so special?
Everything. They do everything.
They do everything, obviously.
They seem to do a lot of stuff, yeah. They're very, I would be biased, but I think they're really cool cells.
And how did you go about studying them? Is this all in vivo? Did you do some cell culturing? Can you cell culture?
So we didn't do any real-time in vivo work because it's really hard to look inside the brain of a mouse while it's alive. So unfortunately, there were some sacrifices made. But a lot of the work that I did was involved in looking at very thin slices of mouse brains with immunofluorescent markers on them. So to light them up in really pretty red, green, and blue pictures.
Which are great for a podcast.
Yeah, I know.
Podcast format.
Trust us, it's beautiful.
Go and read the preprint in the paper. Go and read them.
We're always doing this. We're always picking image-heavy preprints and then saying how wonderful the images are. And it's, we should learn.
It worked really well on my thesis because I didn't really have to write anything. I just put loads of pictures in, but it doesn't work so well. It works well on the paper as well, even though you have limited numbers of figures. And it's really hard to fit everything on one page for a figure. But it doesn't work so well for a podcast, but we'll do our best.
But you've got lots of pretty pictures.
We have got lots of pretty pictures. If anyone's interested in pretty pictures, go and have a look.
Screensavers.
Yep, they make great screensavers. In fact, my favorite is a picture of a molecule called Sonic Hedgehog, which is one of my favorite molecules. It's a picture of Sonic Hedgehog in an embryonic brain in a pattern that looks like a heart. And so I really like it's a good Valentine's Day card cover if anyone's interested.
I know what your girlfriend's getting now.
Yeah.
She gets it every year. She's sick of it.
So yeah, so I did a lot of immunofluorescence as well as doing some, for the people who know a lot about different types of techniques, did a lot of in situ hybridization as well, looking at different markers. And then we also did various different types of cultures as well. We did neurosphere cultures, which we can get onto in a bit. They're like organoids, where you grow cells in suspension. I know you've had people talking about organoids before, but they're very basic ones. They're just cells that grow into balls, and then you have a look at them. And then we did other ones which are called slice cultures, where you take a thick slice of the brain and then try and culture it and keep it alive and see what happens over time. And so we did things like showing that when you take these slices from near the ventricle where the tanycytes are, they don't have any dopaminergic neurons there. But then when you culture them for a few days, you get new dopaminergic neurons, and you seem to get more in brains from wild type mice, these are normal mice, compared to mice where you've removed the NRCam. And then we did some other cool experiments as well, looking at single cell sequencing and things. So this phenotype we were looking at was very subtle. So we tried to approach it from as many different angles as possible to prove that it existed.
Yeah, exactly. It's true.
It's really, really difficult.
Good for us. Everything coincided and all the results told us the same thing. So there you go.
Yeah, it's super difficult looking at things that are subtle changes in science. It's I think it gives you a real appreciation for how difficult science really is, because you do have to look at it from every possible angle just to be certain of what you're seeing. So maybe, Kavitha, you could jump in now with the single cell stuff, because I think maybe I'm horribly wrong, but you were responsible for a lot of that side of things.
So yeah, so this was after Alex, I think he left the lab. So it was after his PhD period. So that's when I came back to Marisha's lab, and she wanted to continue on this work. So we started dissecting out the hypothalamus. So basically what we do is I don't know if you guys are familiar with single cell RNA sequencing, or...
I mean, I'm about to do some. So give me a quick overview so I can learn something that's useful for me.
So what's going to happen is you take your tissue of interest, in our case, which is the hypothalamus, and mainly we focused on the median eminence region. So not the entire hypothalamus, obviously. So the region that we speak about in the paper, and we take them and we dissociate them into single cells basically, and then collect RNA from them and then sequence it basically. And what you're going to get is a map of your different genes that are expressed in every single cell. And yeah, so then they go through lots of clustering analysis, and we have to be honest, we didn't do the bioinformatics ourselves. So our collaborators in John Hopkins, Seth Blackshaw's lab, they are the experts in it. So what we do is we dissect the tissue, make all the single cells, and then send it to them. And they actually do the sequencing and everything for us, send us back these beautiful U maps, which has got these pretty dots, and we can a user-friendly software called Loopfile, which you just go click on your gene of interest, and then it just shows which clusters show your genes of interest, any genes for that matter, just addition molecules or neurogenic genes or astrocytic genes or all of that sort. And then what they do is actually cluster these cells into different populations based on their differential gene expression. And then you can say, okay, this gene is upregulated in this set of population cells. This gene is upregulated in different sets. And that's how we compare between our wild type and NRCam knockout mice. So what we have done in the paper is, we have collected the median eminence region from both your control wild type glutamates and your NRCam knockouts. And we have compared all the different tanycytic genes and different astrocytic markers. And what Seth Blackshaw's lab, Thomas, who's done active sequencing and clustering analysis, he's actually clustered them into the different subsets of tanycytes and seen what markers are upregulated or downregulated between your controls versus your NRCam knockouts. And also he's looked at different neurogenic markers, astrocytic markers. And what we see is a reduction in tanycytes in these NRCam knockouts. And we also see reduction in TRH genes and different genes that are important for your thyroid metabolism. So that was really cool, we thought. And then we have another set of collaborators who did the behavioral studies, and they have shown that there is a modest reduction in light food intake and also a significant reduction in body weight only after about 8 weeks of development. They don't see any difference in body weight or food intake earlier on during the postnatal period.
Really good lesson there in the importance of good collaboration.
I know, they're really good. They did their jobs and they just gave it back to us, and we did our job. So yeah.
I think that's what you want with single cell data.
Exactly.
Here's my samples.
Because it is, to be honest, it is complicated. Hands up, we're not bioinformaticians, had no clue how things are done in background. So they're really useful.
So one of the things that you've both now mentioned, and we should maybe take a little step back and explain, is the NRCam. So could you explain what that is and why you think it's important?
This is what we really need Andy on the call, because
yeah, we should have put Andy in the call.
Like one of the senior authors, Andy Foley, the cell adhesion molecules are like his thing. He's been really keen on for years, looking at all kinds of different systems. And so one of his previous papers that kind of informed the way in which we approached some of this was looking at how, like I mentioned before, sonic hedgehog, but also the sonic hedgehog signaling pathway interacts with an NRCam in the cerebellum to guide how the layers of the cerebellum are set up during development. And so basically, what it turns out that you've got all these molecules that stick on the surface and stick stuff together, but actually they're also involved in modulating signaling pathways and doing all kinds of other cool stuff to change cell behavior, and that actually they're important in different things as well. So while an NRCam is on top of these tanycytes, and it might be sticking them together, there are also a variety of other adhesion molecules on top of the tanycytes. But it's possible that perhaps an NRCam is involved in sitting on the surface of these cells, but it might be involved in like changing how they communicate with each other as well, whether that's for direct contact communication, or it could be through signaling pathways, or other cell adhesion molecules are involved in being at the nodes of Ranvier in like peripheral nerves, so like guiding how electrochemical signals spread across nerves to transmit signals in nerves as well. And so we don't know what the mechanism is for what they're doing, but they seem to be doing something. And so one of the next steps is to kind of like look a bit mechanistically at what it's doing. But yeah, really what it is it's just a cool molecule that sits on the surface and sticks stuff, but then maybe does some other stuff as well.
How did you get on to looking at that? Did it pop up in the sequencing data, or was it before that?
This was really early. Yeah. This was very early. And so I think, like I said, Andy has been looking at these molecules in various different systems for years, whether that's in the peripheral nervous system or in the cerebellum. And so there was some research that shows that cell adhesion molecules are upregulated in certain types of cancers, and seem to promote like motility and proliferation and things that make them more carcinogenic. And like Andy's other work is looking at how sonic hedgehog makes proliferation and differentiation happen in the cerebellum. And so there's a thought, well, maybe one of the cell adhesion molecules we're interested in is involved in here as well. There's been some very, very basic literature from a couple of decades ago that suggested it was present in the hypothalamus. So we wanted to know where it was present and whether it was on the cells. And lo and behold, so Sarah Brown, who was doing this for a master's, tried a few and found that an NRCam just happens to like label mostly tanycytes in the hypothalamus. It might also be on some astrocytes, but it's mostly the tanycytes. And so it turns out it's actually a pretty good marker. It's a pretty good marker for these cells as well. So it was a bit of a shot in the dark that worked out, and then found some interesting stuff based on it as well. Always nice. Which is basic research. This is how it goes.
Yeah. So Marisha's lab does a lot of developmental stuff. So what are you looking at in terms of this? What stage of development are you looking at? Is this something that's on early on, or is this something that kind of more important when we're adults?
The work that we've done in the paper is mainly looking at adult mouse, so between 6 to 8 weeks old mice. But most of the other lab work is like in embryonic chick development, like quite early in development, to see actually how these tanycytes or how these different neurons are specified or set up during development. That's what we're currently focusing on. But the other part is to see in the adults, to see what these cells go on to become and how they are maintained through life and over a life course of things.
Do the levels of the cells fluctuate, or an NRCam actually on the cells fluctuate with development? Or is it quite steady until these sort of cells are needed? Am I asking the important questions? I love it when that happens.
Actually, because like even in our study, if you see, I don't think we see much difference in the NRCam between your wild type and NRCam knockouts in your embryonic stages. It's mainly in postnatal periods, it sets off the difference actually. So I think it plays a major role in terms of stem cell proliferation, differentiation in more the other part of your life than during development. Would you agree, Alex?
Yeah, I think so. So we don't really know about the levels of expression in the cells. I don't think it's really been done, is looking at whether there's more or less expressed at different times. We know that it kind of starts, I think, about embryonic day 14. Mouse have a 21-day development.
Gestation.
Like embryonic development, and then there's gestation. That's the right word. Thank you, Kavita. And then they do a lot of postnatal development as well. So just like humans, except we do a bit of development inside the womb, and then we do a bit of development outside as well. And so our research suggests there do seem to be some differences quite late in the embryonic development. But really the main differences seem to be later on we looked at late embryonic and in adulthood, and we haven't really looked at the time in between because of like it's so hard. It takes up so much time and money to do all this stuff. And so that would be one thing to look at, is when did these effects happen. One hypothesis is that it could get worse over time. Like it could be that you just, like, tanycytes fail to, if they don't have an RCAM, they fail to produce more tanycytes and more neurons and other things that we looked at in the adult over time. And so one thing we wanted to do but didn't get around to doing was looking at aged mice and looking at, like, is there an even more severe phenotype in really old mice? But like, that
was my next question. Yeah.
But then to do that, you have to grow enough mice for 2 or 3 years and then have a look, and so you can't really do that until you know what you're looking for. So you kind of have to have done all the other experiments before you even start doing that.
Yeah.
So it's tricky. It's one of the whole reasons that it's really hard to look at models of aging generally in animals,
especially in mice, I think so.
Yeah.
I have a question.
How dare you?
I know. Well, obviously, I know you can. It's very easy to do things like CRISPR knockouts and RNAi knockdowns in like insects, but in a mouse with something like that, midlife, is it possible to do that kind of, especially in the brain as well? Like, is it possible to do that sort of thing where you kind of knock out or down an RCAM, for example, kind of when they're sort of already more or less fully developed, and see if it makes any difference then or during development? Have you got tools to do that? I have no idea.
Yes, we do. We have all these pre-lock systems, so we can switch on and switch off genes whenever you want. And you can basically drive them with the promoter of your gene of interest, specific to your hypothalamus or any other part for that matter. And then you can switch it on or switch it off and look at the gain of function, loss of function, etc. So it is doable. But the NRCAM knockout mice that we have looked at in this paper is like a normal one. I don't think it can do all these timely knock-on, knock-off effects.
Yeah. So I think it was first developed in about the year 2000. I think the paper it was in was 2001. And so it's not. I think if we told like the senior authors that it was old school, they might disagree, but to me it seems pretty old school. It's a pretty old school way of developing a knockout which is just, if anyone's managed to listen this far and hasn't figured out yet, it's where you remove a gene so it's no longer expressed. And so this is what we did with an RCAM. And so but there are much more modern ways of doing it. So like Kavitha was saying, you can introduce systems that you can be turned on at specific times. You've also got systems like CRISPR that allowed you to like much more accurately target stuff, or you could try lots of different things. But doing it with mice is quite difficult because you need to first of all target the right place, and then they only have a litter of about between like 3 and 11 pups at a time.
5 to 8.
It's usually about 5 to 8. And so then you need to like genotype them, make sure you got the right genotypes, and then you need to breed them. And growing up a colony takes a long time. And like if it takes 10 weeks for them to, it takes 8 weeks. Yeah, exactly. If it takes 8 weeks to get to the point where they can breed again, then it takes a long time. And so setting up a new genetic model takes a long time.
It's pretty much a PhD.
It takes a whole PhD. It's definitely easier now than it used to be, but it's tricky. And so I was quite happy to go ahead and work with this mouse line that we already had, which is another great convenient thing about working on these molecules that Andy had been interested in for years, was we already had the mouse line. I think that's one of the things we actually recommend in the paper, is like you could do some kind of inducible knockout where you could look at, you could turn off an RCAM at a specific time and then see what happened. It would be great to do, but time and money are considerations involved there.
I mean, conveniently, if anyone wants to go and find out more about conditional knockouts in mice, we did an episode on that not that long ago, with a system that lets you turn a gene back on after you've turned it off.
John, maybe you should listen to that.
Yeah, John. Come on, John.
I'm just going to say, I'm not sure it was in mice. I think it was cell-based, wasn't it?
Yeah, that was a cell thing, wasn't it?
Either way, it was really cool.
I think you can probably still you could
probably apply.
Sure, there'd be a way. Yeah,
there'd probably be a way.
I also listened to the average episodes so many times in little chunks while I'm actually editing it that all the meaning of the words just disappears completely. So it might well have been in that episode, but I don't know. I listened to each episode 10 times, but in like 10-second repeated chunks. So
So the obvious question that people always ask when you do things that are not in humans, as if we're all meant to be studying humans all the time, is what's the human relevance here? Do you still find these cells in humans? Are they functioning in the same way?
Obviously, humans have tanycytes, and that's how they function. It is important, like I said, because tanycytes are important for various metabolic functions. So you would assume that when you get old, where you lose all your metabolic physiological functions, there is a reason behind it. So that's one of the reasons why you tend to get all these difficulties when you age.
So they're present in humans. Are you planning to look at different sort of disease states, either in mice or with human samples? Because I mean, the obvious ones I would think of are things like Alzheimer's or potentially Parkinson's.
That's on the list, but not in the immediate future. So the current grant, like I said, the current work in the lab is mostly focused on developmental biology, looking at the hypothalamus development during embryonic stages. I might probably writing some fellowship or something in future. I'm not revealing much.
I think you can keep the fellowship stuff
secret.
secret.
It's only mine. Yeah, but yeah, that's the plan. We want to write because like we don't have current grants funding for mouse work or anything. So we would like to put in a grant to do that, which will be quite interesting because especially with this connection with all these neurodegenerative and metabolic diseases. So it will be cool to look at and also make some collaborations with people who already have these kind of cool mice. Probably that would be an easiest option rather than us reinventing the wheel.
Yeah, from my perspective, as someone who's not in the lab anymore, I can pretty much I can say, oh, I'd love to do all kinds of stuff, and I wouldn't be involved. But no, really, I think one of the cool things that we did was no one had shown that these tanycytes give rise to dopaminergic neurons in the hypothalamus before. There's a couple of different dopaminergic populations there, and we show that one of the populations is reduced when you remove an ARCAM. And if you look in the adult mice, they have a reduced dopaminergic neuron population. And so I don't know if this population is normally affected in something like Parkinson's disease, but it'd be really cool to have a look to see whether there was that effect, but then also, whether an ARCAM did anything. But I mean, I think it'd be really hard because the phenotype is already so subtle, and the hypothalamus under normal conditions seems to produce new neurons so slowly that I don't even know whether you could even look at it at a reasonable timescale. But it would be really cool to have a look at that kind of thing.
Do we know? I mean, we'll have to bring Emma in here, bring the expert in. But do we know if different subpopulations of dopaminergic neurons are
dopaminergic?
Thank you. Do we know if there's sort of subpopulations at play? I imagine or is it just like a blanket? Things go wrong.
The thing is, I don't know specifically about the cell type that Alex is talking about. I was actually quite surprised to hear that they turned, I can't say it, Tanycyte, that they turned into...
Tanycyte.
What is it?
Tanycyte.
Tanycyte, there we go. I think I've been typing Tanycyte in all the producer notes.
You have, and it's been so confusing.
Sorry.
Alex, if you ever do drag, then I think Tanycyte has to be your drag name. I'm christening you here and now.
Tanycyte.
I'll bear that in mind. I'm just make some notes.
Anyway, I wasn't aware that Tanycytes made dopaminergic neurons, but I know that vaguely in the substantia nigra particle factor is the main area that's affected, and there are different subpopulations of dopaminergic neurons. Like even when we do iPSC differentiation, you get different subpopulations, and we look at the one that's closest to the main group that's affected, but there's other little groups as well.
Okay, so it could be having a role,
but it's not I don't know how closely it's located to the substantia nigra particle factor, which is the main area affected in Parkinson's disease. I'm guessing it's not closely. So no. I mean, it could, but I would say Alzheimer's is more likely because that affects the entire brain and just kind of shrinks it. It's crazy.
Yeah, and so I think very little is known about how just generally there's only 3 or 4, I guess, if you include the hypothalamus, which you should. I guess there's only 4 different known neural stem cell niches in mammals, and we know even less about how they work in humans as well. And so there's one thing trying to look at these in mouse and fish and fly models of these neurodegenerative diseases, and other ones trying to look in humans, which live for many, many times longer. And so I think it's difficult. It's so hard. I don't envy people working on neurodegeneration, because it seems like every time you learn something, you realize you know much less, which I guess describes science generally, but it seems to really describe neurodegeneration.
It's really hard to get humans to do what you want them to do too. They do not want to sit under a microscope as you stick their brain under.
No, it's really hard to control their diet and control their sleep cycles, control the medication they take, and account for all those other factors as well.
So let's circle back around a little bit to neurospheres, because they sounded cool, and we like cool sounding things, especially when we've never heard of them before, because I get to be educated, and then I use them for a week and forget all about them again. So what are neurospheres, and why are you looking at them?
Alex, you want to go? Or
I mean, you can go if you want.
So neurospheres are basically 3D suspension culture models. So what you do is you dissect out your tissue and then dissociate them and then put it back in a dish in suspension. And what they do is like they just clump together to form like nice 3-dimensional neurospheres. So similar to your organoid, but in a less sophisticated way, I would say. And what are the advantages? Like you can culture these neurospheres and then you can massage them to show their self-renewal capacity. So like, for instance, if you dissect certain population of tanocytes, they can go on forever, like more than like 10, 15 passages. Whereas like these certain subsets can go only until like 6 or 7 passages. And so that tells you whether they are like more stem cell-like or more progenitor-like. That just gives you an identification of that. And you can also differentiate these neurospheres into your different neuronal subtypes and see if you can, actually see these hypothalamic neurons come differentiating from them. So it's cool in a way. And you can look at all these hypothalamic specific genes and things. The advantage is it's ex vivo and you can look at all genes and like look at the self-renewability, differentiation, etc. So yeah, it's easy and cool.
Which is what we want, easy and cool.
Easy and cool. Yeah, they're easy system to work with, not too difficult to deal with. Yeah. So primary cell culture, basically.
Easy as long as you don't mind coming in all the time and feeding them over the weekend.
Yeah.
I was just going to say, like, how long do you have them in culture? Because like organoids can take up to 90 days sometimes to develop?
So it's not as long as organoids. It's like, say, for instance, from day of dissection, the neurosphere forms within a week. Probably, is that right? Maximum a week should be enough for them to form neurospheres, and you can start massaging them every week or so. And then, you can keep them going for months if you want to.
Are they? So they're proliferating, and they've got multiple cell types and stuff in as well, or is it just one subtype?
Good question.
Because organoids, you get loads of different ones.
They are a heterogeneous population. So yeah.
But then even then, I think while we know they're heterogeneous, but I think the conditions they're in are supposed to like restrict differentiation. So it might just be that they've got they've slightly specialized, but not very much, or become more restricted in what they can do. But the idea is that you're keeping a number of like purish neural stem cells in the middle, which may be like partially differentiating as they split. But then what you do is you take the map suspension, stick them on a flat surface, and take away some of the growth factors, and hopefully make them become other stuff. And so it was so cool looking down the microscope. You've got these amorphous blobs of cells where everything looks the same, and then you make them flat, and they grow into these cool just
neurons and astrocytes.
Yeah, neurons and astrocytes and stuff. And you throw in a bunch of markers, and you can see these pretty pictures down the microscope. And I mean, I complained about the system, but actually I know it's better than other systems. Anyone who's worked with iPSCs and stuff will tell me. I mean,
Emma, I'm waiting for Emma to jump on.
I know. So they're not too bad.
There's a reason I did my current screen in HeLa cells. I'd had enough after 2 years of my differentiation not working.
Where do I find out about the different bioarchive licenses? This CC, BY, CDXY nonsense is driving me nuts.
ASAP Bio have a resource for that.
Ugh, that's your answer to everything.
That's because they have everything you need to know about preprints.
Sure, they probably have the basics, like info on the preprint servers, but what else is there?
There's so much more. Looking to post a preprint, but not sure what different journal policies are? They have a collection to help you out with that. There are meetings around preprints and associated services. If you want to know how preprint adoption has changed over time, there's even a page on that.
And COVID?
They have a big section on preprints and the pandemic, plus some really cool infographics for communicating preprints.
And university policies? Surely they don't have that.
They collect uni policies where possible.
Okay, they do sound pretty impressive, but is it not a bit of an echo chamber?
It can be, but ASAP Bio also engage with people who don't love preprints and have concerns. So we had an excellent discussion on this very topic a couple of months ago.
Oh, is there anything ASAP Bio don't do?
Honestly, no, they're so nice over there. They were so quick to jump in and support this show. It's your one-stop shop for info on preprints and open science initiatives, so head over to asapbio.org to learn more and subscribe to their newsletter for the latest in preprint news. If you want a deeper dive into the world of preprints, then look out for the next recruitment of ASAP Bio Fellows.
Okay, so we ask everyone why they preprinted, whose decision it was, and sort of their experiences around preprinting, because it's the show's name, and we get money for doing that. So how whose decision was driving the posting of the preprint?
I think it was a combined decision. Once we submitted it to the Journal of Interest, we just then thought, okay, fine, we should put it in bioRxiv as well. So it was a combined decision between the authors, I think.
And did you get any sort of benefits from pre-printing it, or was it just to get it out there and make it accessible whilst it was going through the tedious peer review process?
The second one. So just getting it out while we are peer reviewing, however long it took. But luckily in our case, it didn't take that long, was it? It was like only mere 2-3 months. So it was easier to get it there.
Oh, that's quick.
So yeah.
And yeah, this is one of the few we're doing where you've actually published.
Yeah, it's out.
Whilst we're talking to you, rather than a lot of them have now published after we've spoken to them, which lets us ask a second question, which is, did you see much differences between the preprint and the final published paper?
Not much, actually. A bit, but not much. They just asked for a few bits and bobs. Like we did, in terms of experiments, we didn't have to do anything new or any fresh data. It's pretty much rearranging things and saying things in a better way. Basically.
Yeah, we were fortunate to have quite enthusiastic reviewers, which I guess which is not the general experience, but,
which we don't have all the time.
Yeah, you're really selling peer review. This is not what I'm about.
So actually, yeah, they brought us up. I think all the comments they had were pretty good, which is mostly around like changing the way that figures were put together and making sure that the figures were consistent and stuff like that, which is like something that when you've looked at these things for so long, you don't necessarily even think about. And so I think in this instance, it worked quite well. But on the other hand, to big up preprints, like I'm very happy it was pre-printed, because I've been when I first heard about it, I was like, wow, that's such a crazy idea. But I think it's good, because if we hadn't, like if we posted it and then it hadn't got accepted, I mean the work was still there. And so for me, who worked on this quite a long time ago now, I finished my PhD and I submitted my corrections in 2019, and so I didn't know if it was ever going to be published, and then we did loads more stuff with the collaborators, and Kavitha came on and did stuff, but at the time I wasn't sure. And so for me, it was like there was a marker. I don't need this for my career now, but it was really nice to have it there. And I was like, the work I did is actually there, is actually there, and people can see it, which was really cool. And so I'm glad it went up.
And it's really helpful for when you need to go back and read it to remind yourself what you actually did. Because I put a paper out and forget instantly what any of it's about.
Yeah, most people get little lots changed. Yeah,
it doesn't take long.
But here we were fortunate enough not to have much changed from our preprint to this final version.
Yeah, good. Yeah, it sounds like a very nice process, which is what ultimately we want, which is good. And productive and helpful. That's what peer review should be. Quick, helpful, nice comment. Not demanding more lab work. But now we've pushed that.
I think if we tried to publish it after I finished my PhD, like I was very happy with where I finished my PhD, but it wasn't ready for publication. Like there was a very subtle phenotype we'd looked at from 2 directions, and one of them we didn't have enough data on to be convincing. And so to then have like Kavitha come in and do a load of like build up the evidence base of stuff from with our lab, so it's like to really finish stuff off rather than where I left it, and then also to bring in the collaborators, so Seth Blackshaw's lab and then Joe Lewis and Fred Ebling
they have been very helpful and great. Very
like it's like I was convinced there was a phenotype when I left it, but I knew it would be hard to sell. Whereas with all of these other different types of evidence, you're like, okay, no, there really is. We can look at it many different ways and see the same thing, the same subtle thing, many different ways. So yeah, I think I was very happy with where it ended up, and I like to think that was one of the reasons that publication went okay as well.
Which, I mean, that's a really good point, because of course, like you said, not wanting to stay in academia, you don't have that pressure to get a paper, because it's nice to get one out. Everyone wants to share their work with people. That's why we do it. But if you're staying in academia, there's a huge pressure to get your paper out. And when you're working on something where, I mean, Emma works with stem cells that take an age to work with. I'm doing a lot of mouse work at the moment, which takes an age to do. And when you're working on something like that, where you can't do it quickly, you've only got 3 years as a contract to get stuff out. And it's not always possible to do that. So, it really goes to show that the current system it doesn't work anymore.
It is a hard work.
Not really.
To get anything out. You just, like I said, you need to be at the right time at the right space of time. So to get anything done. Because if you have everything set up for you, you just walk in and do your experiments, boom, it works. Otherwise, you just have to set up everything on your own, and that takes forever.
Yeah, I mean, setting up a new lab as well.
Hard work.
It's an arse. It's a massive arse. Having done that now. That leads us nicely into a Kavitha question. So you obviously your plans are to stay in academia, try and get your own lab and do your own stuff. You've done a lot of different things. One of the things, so you used to work with one of my PhD supervisors as a postdoc. You've worked with Marisha. You've kind of bounced around a lot of the different labs at Sheffield. You worked with Emma, and you've worked across a lot of different things too. You've done the thing I like, which is to jump across and get varied experience rather than sticking into one thing. So how have you found that? How has that benefited being a postdoc?
It has actually, I mean, honestly, I'm kind of person who, gets kind of bored after a while of doing the same thing again and again for a long time. I don't know if it's the right thing to say it out loud, but so doing different projects at different periods of time has like actually helped me to focus on what I actually like, in the sense like I have tried different things now. So okay, fine, I've done some bit of basic developmental biology, some translational work, and some neuroscience stuff, etc. So everything has like now pinned me down to this bit where I can use all those experience that I've gained, I think, to see where I want to go next. So, which is useful, actually. I think it's a good exercise rather than just following it up in one go. For me, definitely, it has been useful, as I would say. I don't know about others.
Yeah, I always argue that.
It's a good experience because like it is, you learn new techniques and you learn new ways of, doing experiments and things, right? Rather than just doing your gold standard experiment day in and day out. Because working with Martin's lab was like totally eye-opening for me. I've never done like Western blot in my life before, but it all worked, and it all, so it gave me the right experience that I needed, I think. So it was wonderful. Definitely.
A Western blot, that worked.
No, I was going to say that. Like, what is this magic? Teach us your ways.
So, switching away from that, leaving academia, as someone who is very much going down that path, I imagine it to be this green, rosy, amazing, happy place where everyone skips everywhere, which is probably not true at all.
Not true.
So how have you found that, Alex, as a step? I imagine it's quite different to being in academia.
Yeah, it is. And I think it will be different for different disciplines within academia. But I realized about halfway through my PhD that I didn't want to stay in academia after I was doing my PhD. I'm still very glad that I did it, and it still informs the way that I do my current job, even though I don't do anything to do with neuroscience whatsoever. Everything I do on a daily basis is informed by what I learned doing my PhD. And so I think it was incredibly valuable. And I think that the skills that PhD candidates develop are extremely useful outside of academia as well, even though it's not necessarily apparent what they are until you leave. I think it's very difficult to know when you're inside academia what it's like outside. And so I've definitely had a positive experience. I know that it was a very good decision for me. I've tried not to be too evangelical to my friends who stayed in academia, like all of you guys and many others as well, because I know that different strokes for different folks, like different things work for different types of people. For me, it was great. I struggled through parts of my PhD with like you work an experiment for a really long time and then it wouldn't work. And then you work an experiment for a really long time and it wouldn't work. And so one of the fundamental parts of my PhD work I didn't manage to replicate until 2.5 years in. And then suddenly everything fit together, and the last year and a half went amazing. But like at times it was a struggle because of just the crushing failure of nothing working forever.
My entire second year, when I present my PhD, well, not that I do anymore, but when I present my PhD work, I have a slide, just a single slide to summarize my second year, and it's just all failed, didn't show anything.
I think that's 80% of people. That's how PhDs goes. As far as PhDs goes, I think that's normal.
Yeah, and I just, for me, that way of working wasn't great. Like I know it's important, but it didn't work very well with the way that I motivate myself. And so the first thing I did when I moved out was moved into a kind of I joined the civil service as an operational researcher, which is someone who does like a combination of technical problem solving and then kind of like business management, project management type stuff. It's all a bit nebulous, but I did a role that was involved in like being a statistician and doing data science, and that was a way that I could ask researchy type questions and do stuff, but I'd get my answers within minutes. Like I'd write some code, and when the code wouldn't work, that was fine because I'd know immediately that the code hadn't worked and I could go back and write it again. And so it's just a different way of doing researchy type stuff. And so I do all kinds of things now, but my favorite things to do are still research. It's just different systems, no longer mouse brains. It's other stuff like looking at building a model of recruitment to teacher training, or doing some behavioral science research, looking at social media, different things.
And more direct outcomes. You actually, you produce something quickly, you have an answer quickly, so you can kind of actual influence is happening quicker than
I think that's true. And so I wanted, one of the reasons I wanted to do a PhD was because I wanted to do something that felt like it would make a difference. And there's some hubris involved there, but I think also it's a thing that a lot of people share, is like they want to feel like they've left a mark on things. And also like I did feel like this work that we did is important, and it is important. It's a really important paper. Go and read it, go and recommend it to your friends. Like it's a great paper. The work we did was really, really valuable, but also I like having effect on, like, working in public policy means you can have an effect on things in the immediate term. And so like the modeling I was doing looking at recruitment teacher training meant that we could say, well, actually the numbers aren't looking so good at the moment. Let's try and change the number. Like, how can we try and increase the number of people who are going to be recruited to history teacher training this year? Like, are there something we can do about it now? And so when you see that immediate effect, it feels like you're making a difference. But then again, there's lots of different types of job outside academia where you do different types of things. I know other people who leave and go into the pharmaceutical industry. We all know people who go and work at the dark side of the pharmaceutical industry.
Yeah, we all know.
No, like, and you can do things there as well. Translational research, whether inside or outside academia, can have more of an apparent effect than the basic research, despite the basic research also being fundamental.
I was locked out of my new flat this morning, so I sat and tweeted about how great a new startup in the US is, Arcadia Science. And we're going to probably have those on at some point to plug all about that. But yeah, clearly there's space for a podcast talking about people who've left academia. That was I'm hearing there's a need.
Can I
John,
jump in with something? Yeah,
jump in.
So yeah, I was going to say, so if you, I guess it's one of those, knowing what you know now sort of things, but if you could go back and talk to sort of just deciding to leave academia Alex and be like, right, with the rest of your PhD, you need to spend the time feathering in with skills that we use for down the line. What would you go back and tell him to do? I suspect I have a few guesses for what you might say, but let's see.
Well, realistically, it wouldn't really matter what I said because I wouldn't have done anything different anyway. I think I find it very easy to be motivated by the things I need to do and quite hard to go away and learn new things. And so I was very bad at going away and doing the trumpet practice I needed to do when I was a kid and I was learning to play the trumpet. I'm very bad at going away and learning Lithuanian, which is something I'm supposed to have been doing for years. And so I'm also not great at going. I wanted to do coding and programming and stuff when I was doing my PhD, but there was no immediate use for it. And so I was very bad at like making time to do it. And so that's one of the things I'd like to do because I thought I'd be quite good at it. And turns out I was okay at it as well. And I'd like to do more of it. It's just that my job's moving in a different direction. And so I think that's generally a skill that people who do academic research probably get on with quite well because it's just a way of asking questions and getting a machine to give you an answer. And the syntax of it and like the logical way in which you can construct arguments and questions is quite similar to the way in which we think about how systems work in biology. And so that's something I'd like to do more of. But generally, I think the most important thing wouldn't have been about learning new things. It's about understanding how the skills that we had from our PhDs are useful, whether that's within research generally or outside. Like not just being able to do a Western blot or being able to do different types of experiments, but understanding how you learn, like how you learn different types of experiments or how you go about writing a scientific argument or giving a good presentation, that kind of stuff. Everything everyone has to do as part of their academic work, whether they're a PhD student or at a later stage. And so understanding those things is really valuable. And so something that I do now is I'm on the committee for the Civil Service Systems Thinking Interest Group, which is about bringing civil servants together to kind of learn about systems thinking. And that's how you think about problems inside systems. And so as for government, like understanding the systems at play in fixing policy problems is really important. And it turns out it's really like this systems thinking is basically the only thing you do when you're doing research is understanding systems and how your little part is actually affected by all the other things that fit into it, particularly if you're trying to figure out why an experiment doesn't bloody work. You have to know about all of the other factors that fit in as well. And so, when I first learned about it, I was like, oh, this is great. It's the way my brain works. I want to do more of this. And so one of the things I'm trying to do at the moment is working with some other people to build a systems thinking curriculum for other analysts and others who want to learn about how they can, within the civil service, and how they can kind of bring more of it into their policy making, because I think it's a really valuable skill that researchers develop just by doing the research, but actually is something that's applicable to everyday life.
Good question there, John. I think I've run out of questions. I don't think I've got anything else to ask. I've got 2 bits to end on. Well, so we were a little bit excited when we first started the talk here because we know you both. I mean, we all met up with Alex the other weekend to get drunk together.
For his birthday, not just because we're sad, just to be clear.
For his birthday, yeah. But I mean, I haven't seen Kavitha for years. Since I was in Sheffield, actually. So we were a little bit excited and got carried away with being able to chat to you both. So we kind of skipped over the fun facts you provide. And I really like the fun facts because I think it's nice to get an insight into people who, they're not just scientists, they have lives and they do fun stuff. But also, when we have people on we know, I always learn something. So we had Hannes on a little while ago, and I learned something from his fun fact I didn't know about him. We did a practice episode when we first started with John, and I think I learned something then as well.
That would never see the light of day.
I heard it.
You did. So I do really like the fun facts. And yet again, I I think actually I might have known Alex's fun fact. But so Alex's fun fact was that you used to be an extra for films and TV. It's kind of quite cool, actually.
Wow.
What were you in? Where can we find you?
Well, so I used to live quite close to where Pinewood Studios is, and also there's loads of other studios in London. And so I was part of a children's extras agency, so where it's just provide extras, particularly extra children, to be in the background of films.
Have you
and
got
TV.
any extra children?
Yeah, exactly. Pretty much. So I was in a very bad rendition of Robin Hood that was produced by Russell Crowe, featuring Russell Crowe, and directed by Ridley Scott and Kate Blanchett.
There's a famous tree in that from Northumberland, from where I am, actually. So there you go.
Oh,
We're
right.
connected.
I know someone who was an extra in the beach fighting scene, someone running down the beach with a shield. I know someone from home that was in that scene as an extra. So there you go.
Anybody else here? Anyone else here know anybody in that film? Or trees in
No,
that film?
I'm still impressed that you were in a Russell Crowe film.
I know. Yeah, exactly. We just stopped at that. Russell Crowe, done.
Spent an afternoon with Kate Blanchett. She was very nice.
What?
Wow, nice.
Gladriel.
So my brother did much better. My brother was much more successful at this extraing. In fact, it was almost not extraing. He was on the West End in Oliver and The Wizard of Oz, I believe, and used to understudy for Oliver, and was all very nearly like Oliver on the West End at one point. But
so you didn't push him down the stairs to get the lead role then?
No, but he in a film called The Other Berlin Girl played the son of Eddie Redmayne and Scarlett Johansson, and had lunch just kind of like 2-on-2 with Scarlett Johansson, with him, my mum, and Scarlett Johansson's bodyguard. So that worked pretty well. And yet now he's an accountant. So, you know.
Oh, nice. Good fun fact.
Well, I mean, we just learned loads about Alex. Yeah, so many more questions. So Kavitha's fun fact, though, also really good and kind of, again, creative kind of stuff that we don't often think of scientists doing. So you dance, and you're also on, I can't remember what the cricket team name is, but the Yorkshire.
Yeah, we play for the South Yorkshire Women and Girls Cricket League. Yeah, it's fun. So yeah, we started only last year though. So I've always been an ardent fan of cricket. No surprise there. Asians, Indians coming from a cricket background, but no, always watched it, but never got around to play. But last year we started off. So we have our own cricket team in Sheffield, which is part of the Yorkshire and Derbyshire Cricket League, where the men's used to play. My husband plays for this league called Sheffield Super Kings. And last year we started a women's division. And so we've been really good at it, actually. We won the Plate Cup last time. And this year, this is the second season in, and doing really good. So yeah, that is one of my exciting achievements.
You've got to win again.
I know, we should. There's too much pressure now. So the fun fact is, because your ECB, the English Cricket Board, is pushing women in cricket and women and girls mainly into cricket. They just do all these charity events and things. So last year I had an opportunity to go play for this charity event between Parliament versus Yorkshire, and Ian Botham was the chief guest. I don't know if you guys know Ian Botham. He's this famous English cricketer, and David Gower. And I told Andy that I met both of them because Andy watches cricket a lot. He was well jealous. He was like, I can't believe you went. I just sent them a picture of me and both of them and David Gower. He was like, can't believe you went and met him. So yeah, that was fun. So great. So that's what I'm doing. So we do have a few girls team now. So looking at young girls and boys, started a new young under-9 team as well. So they are playing cricket. So it's more of a community coming together. So, and I'm a committee member of these kids league. So one of them, I sit on the committee. So benches at cricket league for under-9s and under-10s. Yeah, that's few of my achievements outside academia, I would say.
Who would have thought we'd end on such a famous connection there?
I know. Next time, you can see me on this television. I'll wave at you.
Just to see him both have Twitter. Can we tag him in the episode launch tweet?
All I've got is the bloody Robin Hood tree. That's no fun.
Yeah, they've shown you up, Jonny.
And it's not even my tree. I've just stood next to it once. All right, I think that's it. I think we're done.
But we have to say that you've been an amazing host, and I'm sure you'll do good editing, John. Thanks, Emma, for approaching us to do this. It's a good opportunity. Never done this before. Feels like celebrating, huh?
It's been really lovely to have you on and catch up a little bit. I mean, I see Alex all the time, but I don't see you.
I know. We should all have a Sheffield catch up once.
Yeah.
When you come down to Sheffield next. Yeah, cool.
If I ever run away from academia, I'm going to go, chicken farm or something on the outskirts of Sheffield. So yeah, I'll see you back.
Sheffield is beautiful and lovely.
It is.
I
I really miss it.
Yeah,
I miss the hills.
See, I've just, in my entire life, I've just been to two places. One in Chennai, back home in India, and then I moved to Sheffield. I haven't moved out of Sheffield. So this is like my second home.
Well, you know, all of us who've moved out of Sheffield, or I think I'm speaking for all of us who've moved out of Sheffield, deeply regret it. We all want to go back. It's such a lovely, nice city.
It's got the perfect balance of everything, I think. So
I do miss it, and I do. It's nice to see all you guys again, to have us on the show. It's been really, really great. It feels very friendly. Coming in and seeing some friendly faces on the other side, it's not like going into an interview board. So yeah, thanks very much for hosting us and for letting us discuss our work.
Literally, I was like all afternoon. Luckily, my experiment failed this afternoon. So yay for science.
Interesting use of the word luckily.
Exactly. So I had time to like go through stuff and do, actually read the paper once again. So give you the right answers.
Well, I think I've said this a few times, but we had a paper out last year and I was on Radio 4 for it, and they asked me a question, I give them the wrong information. So it's really good to go back and read the paper.
Yes, definitely.
To refresh your memory of what you did before you talk about it.
And you don't have to send out an awkward tweet being like, I'm
so sorry, everyone, I was wrong. All right, I think that's it.
Thanks very much for hosting us and for letting us discuss our work.
It's been really lovely to have you on.
Okay, and that is the show. If you enjoyed listening, then hit that subscribe button for more and leave us a review on whatever platform it is you're listening on. You can reach out to us on Twitter at MotionPod or online at preprintsandmotion.com. Didn't enjoy that? Well, we're all scientists here, so send us your review and let us know what works or what you'd like to hear more of, or less of. But until next time, have a good week.